So, our next speaker for today is a 2023 fall graduate from the College of Liberal Arts and Sciences. They are studying environmental science. Here to present her field study on temperature's effect on microcosm community abundances, please welcome Ms. Lindsey Choi to the podium, or Elsie.
Hello, everybody. My name is Lindsey Choi, and I worked with Dr. Luring and Parker Benz over the recent year in the Department of Biological Sciences at WSU. Our lab focuses on aquatic ecology. Sorry, I'm just getting over being sick. With special consideration to temperature and drought. My team looked at how temperature impacts microbial community abundances after rewetting. In our experiment, we looked at how microbial community abundances respond to the rewetting of soil from an ephemeral pond across the thermal range after an extreme drought period. Due to climate change, we expect that droughts will be increasing in severity. This could be through decreased precipitation, increased temperatures, and earlier and extended droughts. One of the ways that organisms tolerate these conditions is to use ephemeral ponds. These short-lived ponds develop in late winter or early spring. They are refugia for many amphibians and turtles that snack on zooplankton. These microorganisms rapidly repopulate after precipitation events. What we wanted to know is, how is the composition of microbial communities impacted when re-wet across a range of temperatures, and how does the abundance of microbes change in relation to temperature and the length of time after re-wetting? To do this, we looked at what groups occurred in time and their concentrations. To begin our project, we collected 20 10-centimeter soil cores from Bullfrog Pond, a dried ephemeral pond at the Nanesca Reserve, a WSU field station gifted to WSU almost 40 years ago. It is 80% restored prairie and 20% wetland and woodland habitat. These soil cores were taken back to WSU and re-wet with sterile rainwater before placement into their randomly assigned environmental chamber. These chambers were set at a stable temperature of either 16, 20, 24, 28, or 32 degrees Celsius on a 12-12 light-dark cycle. Sampling began 24 hours later and occurred twice a week over the course of 8 weeks. Samples collected were filtered through 1,600 micron nylon strainers before being processed in the FlowCam. The FlowCam 8100 imaged the samples at the manufacturer's optimal estimated efficiency settings. Afterwards, runs were reviewed and cleaned up before categorizing the pictures into their respective phylum to genus when possible. We found there were 3 primary phyla present: Arthropoda, which includes a wide range of organisms from insects to crustaceans. The 2 most abundant genera were Daphnia and Lepistheria. Next, Chlorophyta, which is a green algae. We observed genres Pediastrum, Euderina, and Volvox. This does not really provide a full reflection of the green algae present, as larger filamentous blooms did occur, but they were hard to identify and quantify through our methods. Lastly were the rotifers, a much smaller phylum compared to the other 2. These are filter feeders, and we could not easily distinguish the differences between the 2 classes of Monogonota and Beldoidea. And looking at the numbers within our data, we observed that Arthropoda was the first to appear after rewetting, following Rotifera. Chlorophyta didn't appear until the 2nd week of sampling. Next, we considered who was the most abundant overall and saw that rotifers were highly present, followed by Chlorophyta and Arthropoda. To look at our data, we used statistical modeling of a generalized additive model, which would allow us to see the fluctuations of populations over time. This required smoother terms, which eliminate outliers from the data set to make patterns more noticeable. We use the date of appearance and the day by temperature as our terms. From our results, we can see that arthropods likely use diapause to repopulate after the drought. This is where the egg starts development and then pauses partial the way through until conditions are optimal for it to resume development. When, when repopulating, this showed a significant effect in time from the re-wetting event, and they increased abundance over time. We also found a significant effect in time by temperature on arthropods, where they increased in abundance at higher temperatures. Next, with the chlorophyta, we saw a potentially significant effect from time after re-wetting. This may be skewed as we were able to only identify some of the green algae present, as I mentioned, due to the constraints of the imaging. The algae we did identify are all colonial species, and that may be reflected potentially in the lag seen through the date of the first appearance. Chlorophyta decreased in abundance over time, but they were significantly affected by the time by temperature. This was seen through the decreased abundance to absence at higher temperatures. Lastly, with rotifers, they may have repopulated after the rewetting event through dormancy. This may explain why there was no effect by time after rewetting. Their population took the longest to have its first initial peak in concentration, but increased over time to become the most abundant. There was a significant effect in time by temperature where we observed an increased abundance at higher temperatures. The significance of these results ties into climate change's effect on ephemeral ponds. Stresses on the environment lead to poorer health outcomes for many organisms. Currently, 61% of all freshwater turtles and 54% of all amphibians are at risk of extinction. Their trophic web could be altered by arthropods and rotifers at higher temperatures. Slightly increased temperatures at rewetting could lead to chlorophyta blooms, but at some higher threshold, they could cause the collapse of the trophic web by their absence. By continuing to studying these microorganisms, we can further understand the impact of temperature on these systems. And just to circle back to my research questions, we asked, how is the composition of microbial communities impacted when re-wet across a range of temperatures? Found that arthropods may be early to establish, followed by rotifers, and chlorophyta last. And how does the abundance of microbes change in relation to temperature and the length of time after re-wetting? Over time, arthropods and rotifers will be able to tolerate higher temperatures better than chlorophyta. Thank you so much for your time, and is there any questions?
Quite admittedly, I didn't understand 3/4 of what you said.
Sorry.
And that's okay. So for the yeoman like me, in plain terms, what would you tell me was very significant about your research?
What we saw is that arthropods and rotifers would be the most significant affected by temperature at higher extreme temperatures, say 28 and 32 degrees, where our higher range of temperatures. We would expect if the ponds are being re-wet at those temperatures, at higher temperatures, that they would be the most abundant. So they might outclass some of the other microorganisms that we couldn't identify through our methods.
I apologize if I missed this, but did you take into account the length of time between wetting events prior to taking your sample? And do you think is there any significance to your findings regarding that?
I think what you might be asking is how long between the re-wetting events are the ephemeral ponds re-wet, maybe? Okay, so this bullfrog pond, I can actually show you a picture. The picture I took was the last time it was wet in the fall, and since then it didn't refill until this summer, actually. In June, at the end of June is when we saw it refill. So it had been dry for quite a long time. So we expect that these temperatures actually can play a large part in how the ecosystem interacts with the larger organisms, such as the frogs using the pond for reproduction, because they need these microorganisms for their tadpole seat.